don't know the setting of this extract, but essentially what it is that all the body's members, except for, let's call it the GI tract, are sort of talking to the GI tract. And everything else is saying, we're important. They rebelled against the belly because they accuse it of not doing anything important. And somewhere down here, the belly replies, so it receives the food which you, the rest of the body, lives upon. And the belly is the storehouse and the shop of the whole body. And I send, don't forget this, he says, or it says, I send it to the rivers of your blood, this is the nutrition, to everywhere, to the core, to, I'm sure he means by that, maybe, you know what that is, the heart, the seat of the brain, and everywhere, essentially. So everything needs this nutrition, and the one that sends it around, so I'm very important. So I think that's just of interest. If anybody wants to see it again later on, I can definitely look at that. So to get to the science of this, where we left it last time was that the food, which was mixed with saliva, passed down the esophagus and through the lower esophageal sphincter. We're talking here smooth muscle. The sphincter is smooth muscle, which is under control, which allows it to open up at the appropriate time. We don't want it to be open too much, otherwise the stomach contents, as we saw, might go back up again into the esophagus, and that can cause problems. So just a bit of nomenclature here. The stomach is described as a J-shaped bag, shaped like the letter J. It's smooth muscle, and the wall has the usual layers that we talked about in the first lecture, with mucosa, submucosa, and then different smooth muscle layers. The upper region of the stomach is called the fundus. The main part of the stomach here is the body, or the corpus of the stomach. And then the lower region, approximately the lower third, is known as the antrum. There's another sphincter-like region at the exit from the stomach called the pyloric sphincter. So you've got regulation over the entry and also the exit of the food and mixtures with various secretions into and out of the stomach. And then this leads into the first part of the small intestine, the duodenum, and is joined by the duct and pancreas. And we'll talk about that later on. What I want you to do for a moment is to imagine that you could somehow be miniaturized and you're inside the stomach. And you're standing there and you're looking around you, what would you see? What you would see under your feet and all around you is a sort of shiny, glistening surface. And this is studded with what would look like little pinpricks or little holes. Now they're not really holes. What those are what are called gastric pits. So let's say initially you're somewhere in the main body of the stomach. And these little, called holes for a better word, are the entry points or gastric glands into the lumen of the stomach. You're standing in the lumen, and here in the wall is an example of one of these glands. So this is what you'll be seeing as these little holes. As you can see, there are multiple cell types here, and I want to just mention these because the products that they secrete are extremely important in the function and the overall importance of the stomach. So let's start with the surface. The inner lining of the stomach, the one I said you would see as a shiny surface, it looks shiny because it's covered with a layer of mucus. And this mucus has got a lot of bicarbonate ions mixed in with it. And this provides a barrier which helps to prevent digestion damage to the stomach itself. It has to be very well protected because, as we'll see, the secretions that it produces would, if nothing stopped them, eat away at the wall of the stomach itself. The gastric pit is this wider opening. Here's the gland going down in the mucosal layer of the stomach wall. The cells which produce the mucus. Number 1 here, the surface epithelial cells which produce the mucus and the bicarbonate. A bit lower down, number 2, there are neck mucus cells. This is the neck region of the gland. And then the ones I have coloured here, number 3, are stem cells. And these can give rise to all the other cell types. And there's a migration of these stem cells, their products, as they move either up or down. They differentiate into these other cell types. There are other mucus-producing cells, number 8, called goblet cells, which tend to be deeper down in the gland. And then other ones here, the ones I've got in green, no particular significance for colour or exact location. These are called, number 4, the parietal or oxytic cells. And we'll be coming back to those in more detail. They're very important. They produce hydrochloric acid, which, when you think about it, is a really strange thing for the body to produce, but there are reasons for it. And they also produce something called intrinsic factor, which was mentioned very briefly in blood lectures. Now, I'll mention that. Numbers 5 and 7, these are hormone-producing cells, heterochromatin-like cells, which produce histamine, and D cells, which produce somatostatin. Again, within the wall of the gland, number 6 here, something called chief cells, which produce pepsinogen. And pepsinogen, as we'll see, has a role in initiating digestion of proteins. Remember that the salivary glands produce what enzymes the salivary glands produce? Amylase, which initiate the digestion of carbohydrates. Okay, so here we've got something we'll talk about that later. If you move down into the lower portion of the stomach, the antral region, so this lower third down here, again you've got gastric glands, but they look somewhat different. Again, you've got a lot of mucus-producing cells, important for protection. You've got stem cells. I ran out of colours, but these are different. These green ones are different from the previous green ones. These green ones are called G cells, and they produce the hormone gastrin. And again, we'll come back to that. There are other cells which produce somatostatin, and more goblet cells down here. So notice no hydrochloric acid producing cells in the antral portion of the stomach. No G cells which produce gastrin in the main body of the stomach. Maybe you've got one here and there. So in general, there's a separation. Hormone production, the gastrin production in the lower third of the stomach. Acid is higher up. So, what are the functions of the stomach? Fairly straightforward summary here of what it is. But it may be a bit surprising to the one I put at the top, the one that's really crucial for life. The only one is the secretion of intrinsic factor. Let me just remind you why that is. It's necessary for absorption of B12. Why do you need B12? You need B12 for normal red blood cell production. B12 is a big molecule. It's not very easy to absorb just by itself. But what happens is that you get your B12 from the diet. You get the intrinsic factor coming from the stomach. As the food goes down containing the B12 binds with intrinsic factor. And at the bottom end of the small intestine, there's special transport systems which allow for absorption of B12. And if you don't have it, you get a type of anemia called pernicious anemia. So that is, it's really nothing to do with digestion per se, but it is a key function of the stomach. The other ones, important, but not essential for life, really. Temporary storage of food, destruction, number 3, of most, but not all microorganisms. And this is one of the advantages of having this strong acid solution in the stomach. it will destroy many of the pathogens that might come in with your food. Number 4, stomach has got smooth muscle in the wall that contracts, relaxes. It can mix the food with the gastric juice. It can move it on at the appropriate rate, and so on. Enzyme pepsin initiates digestion, and I'm going to come back to most of these in a moment and talk about it in more detail. And then the final thing is, after all this mixing and some digestion and so on, you want to deliver the food at the appropriate rate, not too fast, not too slow, to the small intestine. You don't have this figure. What is it? The Great Lakes. Any of you know why I put up the figure of the Great Lakes? Okay, the key thing is this little dot here. So this is, what is it, Lake Superior, Michigan, Huron. You don't need to remember this, you're not going to Huron and Ontario. Anyway, so here between Lake Michigan and Huron, there's a little island called Lake Mackinac or Michilimackinac, something like that. And the reason why I'm mentioning this is that the story of the stomach and how we got our knowledge about how the stomach works has got a Canadian connection, and I think it might be of interest. So just a little bit of sort of historical background. We're talking here back in the 1800s, and there are 2 main characters that are relevant to this story. And one of them is somebody called Alexis Saint-Martin, and he was born near Montreal, and he became a voyageur, working the fur trade. And what happened to him, at some point he was on this little island, and he was shot at close range by some other person in the group, by accident. And he wasn't expected to live, Saint-Martin. He had a big hole in the wall of his abdomen and through his stomach too. And it was very, very bad. But luckily for him, also on this island, the other main character in the story was William Beaumont. And William Beaumont was a surgeon in the American Army, stationed there. And so Beaumont was called over and he rescued Saint-Martin. He saved his life. Unfortunately, although Saint-Martin he recovered, but essentially his stomach didn't. There was still a hole through the wall of the stomach and through the wall of the abdomen. This is back in 1822, so I imagine the surgery was pretty primitive at that time. So whenever Saint-Martin ate food, it would come out unless, there was a padding bandage or something to hold it in. Beaumont apparently was a very ambitious person, and he saw here an opportunity for him to do some studies on the function of the stomach. And so he somehow persuaded Saint-Martin to live with Beaumont in his home, and Beaumont would feed him and look after him and all the rest of it, with the understanding that he would do experiments on him. Soon there were no informed consent things around at that time. So Saint-Martin did agree to do that, and Beaumont did various experiments. And the sorts of things that he did was to put different types of food, a lump of meat, a lump of something else, he tied them with little silk threads, he put them in through this hole into the stomach, he would leave them there for different lengths of time, he could then take them out again, and he could follow the time course of breakdown. And he showed that different sorts of foods were broken down, but at different rates, depending, was it meat, was it rice, was it something else. He also measured the temperature in the stomach, because I think maybe some people thought that the food was cooked in the stomach, maybe really hot in there. He showed no, that wasn't the case. It was 100 degrees Fahrenheit, body temperature essentially, so the food didn't get cooked. He could also look at the amount of secretions and some idea of what caused the secretion rate to change, so that if Saint-Martin was shown some food, smelt it, then the secretion rate would increase. If Saint-Martin got angry, upset, which surprisingly he often did, but again, this could interfere with rate of secretion, or it could change the colour, which is a measure of blood flow inside the stomach. Beaumont also shows that the secretions were very acidic. I don't think he was able to show that it was actually hydrochloric acid, but he did know it was a very acidic secretion. Now, what I'm sort of leading up to here with that background, I'm going to go to the science in a minute, but I have a poem here, which was back in the mid-1990s. It won a prize at a poetry contest. It won third prize at the contest. This was somewhere, I think, in Michigan, the state of Michigan. I just quickly like to read this to you, but before I do, I should maybe add that this was a contest for bad poetry. So it's a terrible poem, really, but it's sort of, there can't be that many poems that are written about holes in the stomach. Okay? Anyway, so it goes something like this. It was at Michilimackinac in 1822 that a momentous medical event occurred, which I will relate to you. A strapping French-Canadian lad, Alexis Saint-Martin by name, in one historic moment sank, was blasted into fame. He was checking out his trapping lines when a musket fearful crack hit him midsection, as I said, at Michilimackinac. That fateful gunshot laid him low, they stretched him on his bed. His stomach wall quite blasted off, but he was far from dead. Alas for poor Alexis, his stomach would not heal. Poor lad Alexis, his stomach could not seal. Then enter William Beaumont, a surgeon most superior. He peered and poured and peered some more at the stomach wall anterior. Oh, lucky William Beaumont, what scientific luck, privy to gastric mysteries through a passage to the gut. Intrepid William Beaumont, not many so would yearn to probe in such an orifice and watch the contents churn. To quantify, to qualify effects of gastric juice, digestion up to then had been a subject quite abstruse. Alexis wished that he had not for such fame been selected. No bite he ate, nor fruit nor meat passed through him undetected. Heroically, the doc observed through the fistula or passage how various food that Alexis chewed was converted into gassage. He checked out food both cooked and raw and also cold and hot. Dr. Beaumont, a great success, but Alexis, unfortunately not. But Alexis was a hero too, and almost forgotten name, his stomach, contributed immeasurably to Dr. Beaumont's fame. Together as a team, they solved digestive gastric mystery, and so together are enshrined in Michigan's gastric history. I gather that Beaumont died at the age of about 50, some 58, wasn't quite old, but Alexis Saint-Martin lived into his 80s, probably some poetic justice there, and he's buried in a small village 40 miles from Montreal. Okay, science. Acid. Why all this acid? How is it produced? Why is it produced? How is it regulated? That's what I want to talk about next. So, figure number 27. We're looking in more detail at a couple of cells which produce acid. And so we're looking in the wall of one of these gastric glands in the main part of the stomach, and these are the parietal or oxygenated cells. At the top is the resting cell, and the bottom is one which is actively secreting acid. What strikes one most about these cells, I think, are two things. One is that they are full of mitochondria, lots of mitochondria in them. The other thing is that certainly in the active, or especially in the active cell, there's a large surface area on the apical side of the cell. This is the face next to the lumen gland, so where the secretion is going to. Call this sort of the back side or the back door of the cell, next to the blood supply. This is the front door of the apical side, where the secretion is going into the lumen. So these two things, lots of mitochondria, large surface area, I think suggest that there's some active processes going on. And indeed there are. The secretion of hydrochloric acid, particularly the secretion of hydrogen ions, occurs against an enormous uphill gradient. If you measure the concentration of hydrogen ions, either in the blood or within the cell itself, their concentration is very low. If you measure the concentration of hydrogen ions here in the lumen, it's very high. The gradient, exact number varies depending where you look, but it's in the order of a few million-fold uphill gradient for hydrogen ions to move into the lumen. If you collect pure secretion coming from these cells in the gastric lumen, the pH can drop as low as about pH 1. And this is extremely strong. It's not strong enough to eat away holes in the carpet and a cat or something that brings up the stomach contents. You don't want to let them sit for too long on the carpet because it can eat away at the material there. So the question then is why and how is this happening? Let's look first of all at the how it's produced. This is figure number 28. So what we're looking at here is this same parietal cell, but drawn in a more star-like fashion. So you've got the blood on your right-hand side, this is the back side, the basolateral side of the cell, this is the apical or luminal side, and here's the lumen of the gastric gland. So the question that one needs to ask is, how do hydrogen and chloride ions get into the lumen here? Well, the starting point for this, and I should say that if you look in different books, you might find the details are slightly different from the version I'm giving you here. Different books have slightly different versions, but I think this is basically correct. The starting materials are simply CO2 and water. These can come from blood, from extracellular fluid, or maybe produced within the cell itself. The enzyme carbonic anhydrase, A, is present in these parietal cells, and under its influence you get the formation of carbonic acid, and then this will dissociate to hydrogen ions and bicarbonate ions. The hydrogen ions move across the apical membrane by a very interesting and important hydrogen-potassium ATPase, a hydrogen-potassium pump. Where do these pumps come from? They come from these tubular vesicles here. Can't see it very well, but there are little black dots here, and I've just redrawn it. It's the same thing here. These tubular vesicles in these cells have in them, they're essentially a storage depot for hydrogen potassium ATPase. Notice hydrogen potassium, not the one that you're probably more familiar with, sodium potassium. This is hydrogen potassium ATPase. And in the resting state, these vesicles just precipitate, and the ATPase is not really doing it. But when the cell becomes active, these vesicles move to and fuse with the apical membrane. So this is doing two things. It's inserting the hydrogen potassium ATPase in that apical membrane, activated, it can do its job, and it's also increasing the surface area of that apical membrane, making it more effective and more room for this active transport to take place. So hydrogen comes out, potassium goes the other way, and there are potassium channels that allow the recirculation of that potassium. What about the bicarbonate that's produced? You can't allow that to accumulate within the cell. So every time the carbonic acid associated with the hydrogen which goes out of the front door, you have a bicarbonate ion which you have to do something with. You can't allow it to change the pH inside the cell. So what happens is that you get rid of it out of the back door, and it goes into the blood through this bicarbonate chloride exchanger present in the basolateral membrane. Now you might say, well, okay, I understand that bicarbonate ions cannot be allowed to accumulate inside the cell because you're going to upset the pH. But you just put them into the blood. Isn't that going to alter the blood pH? This is known as the alkaline tide. So whenever you have hydrogen ions going into the lumen, you inevitably have this alkaline tide in the blood. And I would ask you just to accept that for the moment. I would say that it actually turns out not to be a problem, but I'll explain why that is when we move on a bit lower down the track. Just remember that it happens. So now we've got these chloride ions, and they go out of the front door of the cell through chloride channels. And so you have hydrogen chloride going into the lumen. So that's briefly how it happens. Why? Why does it happen? Well, I can think of one of the reasons, and that is, it sounds a bit funny to say it, but it's protective. Yes, it's potentially very destructive, but at the same time, it has some protective, or confers some protective advantages in destroying bacteria or other things too. But another reason for having hydrochloric acid is to do with the digestive enzyme pepsin. Pepsin is produced as a precursor, an inactive form of pepsinogen. This is what's actually produced in the chief cells in the stomach. And this idea of producing proteolytic enzymes, which is what pepsin is, in an inactive precursor form, is something that we'll see again and again as we go further down the track. And the reason for this is that you don't want active proteolytic enzymes sitting inside the cells where they're produced, because they would just start digesting things around them. So they're produced in an inactive precursor form from the chief or peptic cells, which is, we'll talk about ACH's role, it stimulates them. And then you need hydrochloric acid from the parietal cells to activate the pepsin. So you need a low pH in order to convert the inactive pepsinogen to the active pepsin. Once some pepsin has been produced, that can act in an autocatalytic manner, in which it feeds back on its own production, this one here, and can stimulate further activation of pepsinogen to pepsin. What does pepsin do? So it's a proteolytic enzyme. It acts proteins in the food, because when it's activated, this activation will take place only in the lumen of the stomach, and only when food is present to act as a substrate rather than acting on the stomach itself. And proteins are broken down by this pepsin at a low pH. The optimal pH for pepsin is rather unusual. Most enzymes prefer to work at neutral or maybe even slightly alkaline pH. Pepsin requires a pH where somewhere in the sort of 2, 3, 4 pH range. If the pH goes above, I think, about 5, then pepsin actually stops working. So it's a partial breakdown. Proteins are broken down to peptides and some amino acids. So that's a little bit about how and why. What about regulation? This is something that's always very important to consider for any function, any activity that's going on in the body as a whole, but GI tract. How is it regulated? Regulation of gastric secretion is complicated. There are multiple control systems. And I'm going to try and simplify it a bit by just focusing on the one that we know most about, which is the secretion of hydrochloric acid. Probably the pepsinogen and the intrinsic factor and these other things have similar sorts of regulation, but most of the work has been done on the acid. And I'm going to do this in a sort of two-step way. I'm going to ask and answer the question, first of all, is what is it? What factors regulate secretion of hydrochloric acid? And then move on to the next one. We'll say, how do these different factors work together in a relatively real-life situation? So this is the first question, figure number 30. What are the factors? And you can see the main ones listed here. Nervous control. We're talking here about smooth muscle. We're talking about glands. So we're talking autonomic regulation. And the parasympathetic, through release of acetylcholine, will act on muscarinic receptors on the parietal cell and will stimulate. Plus signs means an increased stimulation of secretion. So that's one thing, nerve. There are traditional hormones that also play a role. Gastrin. Gastrin comes from the G cells in the antral region of the stomach. Gastrin is a traditional hormone. A traditional hormone, when it's released, goes into the blood, goes around the body, comes back and acts on its target. The gastrin from the G cells into the circulation, around the body, comes back, and one of its targets are the parietal cells. And gastrin is also a stimulator of secretion. In the middle here, we've got two local hormones. Histamine from the enterochromatin light cells and somatostatin from the D cell. Histamine, potent stimulator of gastric secretion. So ACH, gastrin, histamine, all potent stimulators. You probably need something to balance that. If you've got lots of stimulation, you need something to have the opposite effect. And that is somatostatin. Somatostatin, I think of it as the great inhibitor. As far as I know, can't think of anything offhand which doesn't fit in with this. But I think everything that somatostatin does, it inhibits. It's always got an inhibitory action. You're going to come across somatostatin again in your endocrine lectures from Dr. Harvey. And certainly one of the things he would tell you, I know, would be that somatostatin has an inhibitory action somewhat in his field of lecturing. So I won't talk about that now, but that's maybe just one way to help you remember. So that's what the factors are. But as I hinted, it's not quite that straightforward. When we ask the question, when you eat a meal or when you're thinking about food, what does that do to these various factors? How do they interact? And this sort of rather complicated figure number 31 tries to answer that question, at least to some extent. It's not the whole story, but I think it's enough for now. So what are you looking at here? We're looking at this outline here is the stomach. Below the dotted line you have the antral region, and above it you have the main body of the stomach. In the main body of the stomach you have parietal cells which produce the hydrochloric acid, and this is what we're focusing on here. So, if you take these four factors here, that's the same factor that is really added much, but a few more arrows here and there. Take these four factors and see how they are working over here. So in blue we have the parasympathetic nerve. The term vagus is often used interchangeably. If you remember, the vagus is the 10th cranial nerve. So it is a parasympathetic nerve. And it's the main one that innervates structures within the abdominal cavity. So some of the fibers from this vagus parasympathetic nerve go directly to the parietal cell. They release acetylcholine and they stimulate secretion. So that's really what we were talking about here, direct innervation, direct action of ACH on the parietal cell. But you can see that that's not the whole story. Some of the parasympathetic fibers will act on G cells up in the main body of the stomach, and they inhibit the release of somatostatin. Why do they inhibit it? Well, we know that somatostatin inhibits the parietal cells. So it wouldn't make sense for ACH to stimulate the release of an inhibitor, because its overall effect is stimulatory. You keep that in mind, that activation of the parasympathetic nerve always increases acid secretion. You need to have inhibition, so two negatives make a positive, inhibition of an inhibitor. There are other fibers which do a similar sort of thing down in the antral region. There are D cells down there, and again ACH inhibits their release. And in this case, what's happening is that somatostatin otherwise would inhibit gastrin release. Gastrin is a stimulator. So two negatives, ACH inhibits an inhibitor, which means that it's stimulating gastrin release. There are also direct nervous controls over gastrin release. And notice that I put this substance GRP, stands for gastrin-releasing peptide. This is one of those situations where there probably are non-cholinergic transmitters of what is basically a cholinergic nerve. The vagus, the parasympathetic, basically it's a cholinergic ACH-releasing nerve. But there are exceptions to that, where other transmitters may be released either instead or in addition to the traditional ones. And this is one example, this gastrin-releasing peptide. Regardless of the details, the end result is that if you inhibit somatostatin, you get more gastrin. You can do this through nervous control, and you can also alter gastrin release depending on the type of food, the contents of the stomach. So there are messages coming not only from nerves and from hormones, but also from the local environment. Proteins have started to be broken down to peptides and amino acids. And certain peptides particularly can stimulate the G cells and give you gastrin. Gastrin, additional hormone, goes into the blood, around the body, comes back again, can act directly on the parietal cells, which would be the gastrin action that we're looking at here, acting directly. And it can also act indirectly through stimulating release of histamine. Histamine can act directly, okay, that's what we showed here. And one that I haven't mentioned was that somatostatin can also inhibit histamine. So So as I said before, I'm pretty sure that this is not the complete picture. But the purpose of this is to give you a general idea as to how these different control systems, this is what they are, and this is saying a little bit about how they interact. And you might wonder, why does it need to be so complex? I guess the more, in a way, the more steps, the more interactions you have, the greater opportunity there is for finer control. And we'll look at that in a bit more detail at another point. Okay, having got this very detailed idea, I want to now sort of take a metaphorical step back and look more at the overall picture. So these are what the factors are. This is how they work together. But what's causing these things to be released? When are they released? And when do they stop being released? In other words, when you eat a meal, how does all this work? So figure number 32 tries to put that together. Now what you really need to have is all this detail tucked away inside the stomach here. That's far too much. So I have simplified this just to the point of having parietal cells producing hydrochloric acid, presence of food, some G cells, but I haven't put in all the details. So what is this telling us? The regulation of gastric secretion is traditionally divided into 3 phases. You've already come across this idea of describing things in phases. When I talked about swallowing, it makes it easier. But I talked about an oral phase, a pharyngeal, esophageal phase, depending on where the food was at the moment. Same sort of idea here, different terminology, but people talk about the cephalic, gastric, and intestinal phase of gastric secretion. So this is representing the site. Cephalic is to do with the head. This is stimuli that are activated in before you take food into the body. Gastric phase, what happens when the food gets into the stomach itself, and then the third one, intestinal, when it moves on into the intestine. And people have described the cephalic phase as being like switching on the engine of the car. You're getting things going. The gastric phase is the accelerator. You put the foot down and things speed up. And then the intestinal phase is when you put your foot on the brake. You don't need or want or shouldn't go faster and faster all the time. You've got to slow down at some point, and that's what the intestinal phase is doing. So you're starting things off, switching on, accelerating, and then braking. Okay, in physiological terms, what's involved? So as it says here, the cephalic phase, which is anticipatory, it's getting things ready. Your body is saying, yes, I see food, I smell it, I hear noises associated with preparation of the food. Or when it actually does go into the first part of the body, tasting. These are things which tell your body that from long experience, any moment now, yes, food is going to arrive in the stomach. So get things ready, it says. Don't wait until the food gets there and then start rushing around and trying to secrete things. Get it going now. So these things, the sight, the smell, etc., work through vagal nuclei in the brain, increasing parasympathetic nerve activity and stimulating this anticipatory secretion of juices. Now, you don't want it to be too much, because you don't want a lot of acid in an empty stomach, but it's things that are just sort of ticking over. But once the food has moved down into the stomach, you move into the gastric phase, and this is the major one. It makes sense. Once the food is actually there, and the body knows it's not being deceived, somebody wasn't just playing with it and said, "Oh, smell this, isn't this nice?" and taking it away again. Food really has come. Now's the time to secrete even more. So the presence of food, distension, so that could be mechanical signals, distending, activation, in other words, intrinsic neuroplexuses, or it could be messages going up the long reflexes, up the brain and back down again, the presence of certain types of food, particularly peptides we saw were effective in releasing gastrin. Gastrin released goes into the blood, round the body, comes back again, and although I'm not showing it in map figure number 13.2, all these things that we had in the previous figure 31 would also be playing their role. So this is just a simplified version that you have the presence of the food in various ways, distension, mechanical, chemical, release of these various hormones, local hormones, local transmitter releases. All of these are exciting further acid and presumably other components secretion. But as I say, once the food has been broken down and mechanically, chemically goes into the small intestine, there may be a further small increase to sort of finish off the food that's maybe too slow and isn't coming down quickly enough. But this phase is mainly putting on the brakes, inhibiting. It's due to the presence of the food down here, but there's quite a bit of controversy about what actually is sending an inhibitory message back to the stomach. I've put here a couple of possibilities. GIP, which stands for gastric inhibitory peptide. Gastric inhibitory peptide. I think it was on one of the lists in an earlier figure. This is where it got its name from initially. Released from the wall of the first part of the small intestine. Maybe other hormones such as secretin, and maybe the most important one of all, this one is a question mark, ones we don't yet know about. There's a whole family of these things, probably, that are released, go into the blood, and work back in an inhibitory manner on various cells in the stomach. There may also be local nerve reflexes, the presence of the food here, whether it's distension or whether it's chemical, particularly these ones here, the presence of acid or fat and protein digestion products, changes in osmolarity and so on, in the first part of the small intestine, could also activate local nerve reflexes, which then feed back in these plexuses that we talked about, having an inhibitory effect on gastric secretion. And I think I'll leave it there and go on to the motility aspect. And